Tony Listro is from Foster Delivery Sciences, and we'll talk about polymer implants and processing techniques. He's Vice President of Foster Delivery, CDMO, in the Northeast, doing implant work, and responsible for continued research and development of new melt extrusion technologies for Foster and for clients. Tony's an expert in the area of polymer materials and processing, and has worked on blending active ingredients with polymers for various drug delivery applications, including oral and implantable dosage forms, for more than 15 years. Tony holds a BS and MS in plastics engineering from University of Massachusetts and an MBA from University of Massachusetts Amherst. Tony, we'll let your presentation start right now, and we'll take some questions afterwards.
Hi, my name is Tony Listro, and today I'm going to speak to you about polymer materials and processing techniques that are used to manufacture long-term implantable dosage forms. Today I'm going to speak about long-acting implants based on polymer materials with a focus on implantable dosage forms that are made by polymer extrusion techniques. I will review the types of polymers that are used, discuss manufacturing considerations and the process options that are available, along with a few case studies. I'll then summarize my presentation. So what are long-acting implants? Why are they used? And what are the mechanisms for drug delivery? Nexplanon is a good example of an implant that has been on the market for many years. It's a long-acting implant that is used for birth control. Nexplanon is a sterile implantable rod that is dosed in the patient using a disposable applicator. Implantation takes less than 30 seconds. Nexplanon is implanted in the patient's arm, provides pregnancy protection for up to 3 years, and is 99% effective. So it's a good example of a long-acting implant. Why are long-acting implants important? They provide localized, site-specific drug delivery that improve the effectiveness of the drug and minimize systemic side effects since the drug is administered locally. These dosage forms reduce the need for repeated injections and enable patient compliance since the patient doesn't have to frequently remember to take their medication daily, as in the case with oral contraceptives, for example. The graph shows an example of the peaks and valleys that occur with conventional dosage forms, shown in dark blue, compared to the steady drug release that can be achieved with a well-designed long-acting implant, shown in green. Polymer-based long-acting implants deliver drugs through one of two mechanisms: diffusion-controlled or an erodible matrix. Diffusion-controlled delivery involves the diffusion of the drug through a polymer matrix over time. Through the process, the polymer remains intact while the drug is gradually deployed to the therapeutic site. This can be achieved either by the drug migrating through the polymer matrix or loading the drug at a very high level, enabling particle-to-particle contact. Drug delivery through migration depends upon the mobility of the drug in the polymer matrix. In highly loaded systems, drug delivery relies on the percolation of the drug, where the drug particles are in contact with each other from the core to the surface of the implant. Drug delivery in an erodible matrix involves the encapsulation or distribution of the drug in an erodible polymer, such as a water-soluble or bioresorbable polymer. As the polymer erodes in the body, the drug is released. Here is a list of approved long-acting implants that are on the market. I'm not going to discuss each of these. However, you can see the list covers a broad range of indications, from cancer to contraception, ocular, opioid dependence, and more. It is interesting to note the range of dosing frequencies range from 1 to 3 months for AZ Zoladex to 60 months for Bayer's Jadel. This demonstrates the flexibility of this technology as a drug delivery platform. Here are some advantages of extrusion technology to manufacture long-acting implants. It's an economical process. It accommodates a wide variety of raw materials. Multiple dosage forms, such as profiled shapes, rods, pellets, and films, are possible. It offers improved drug stability since the API is embedded in a polymer matrix, and the polymer can act as an inherent package for the API. The process can be easily instrumented to obtain online process monitoring, which is in line with the FDA's PAT initiative. It's capable of handling both hydrophobic and hydrophilic drugs. Extra high loadings are possible, up to 60% by weight. And it has an ability to maintain the API's crystallinity. Now I'll discuss some of the types of polymers that are used to make long-acting implants. So what are the attributes for implantable polymers? Well, they're synthetic. They are inert to the body. They're biocompatible. They have a low bioburden, and they're endotoxin free. They can be processed using conventional melt processing techniques. They're sterilizable using gamma and ETO. It's preferred if they have a tailorable chemistry where the molecular weight, copolymerization, and end group modification can be tailored. They're compatible with drugs and the extrusion process. So there are a couple of options in terms of implantable polymers, and in the broadest of categories, they're either bioresorbable or they're biodurable. With bioresorbable polymers, you can avoid the removal of the implant at the end of the therapy, as it's absorbed or metabolized in the body over time. These are useful in non-structural drug delivery, meaning you wouldn't want to use it in a device that's doing some other mechanical function. These types of implants may be difficult to remove, so if there's a concern with an adverse effect or adverse interaction in the body, these can be very difficult to remove, and in that case, you want to use a biodurable polymer. So biodurable polymers are easy. They are easy to remove, and you can remove them intact if that's necessary. They typically can achieve longer durations. They are used in structural drug delivery, meaning again, where the implant also serves some other mechanical purpose. And they maintain their mechanical integrity during the implantation period. So the bioresorbable polymer selection, there are 4 broad types: polyglycolides, polylactides, copolymers of these, and polycaprolactones. The graph on the right here shows the length of absorption time in the body for these different polymer categories. And really the width or the breadth of these bars in the graph are really an indication of the molecular weights that are available for the different polymers. Some factors that affect drug delivery and biodegradation: the chemical structure and composition, the molecular weight and distribution, morphology, meaning the degree of crystallinity, for example, or branching of polymer chains, the site of implantation, so where it is implanted in the body, the part shape and design, since certainly surface area will affect drug delivery, the mechanism of hydrolysis, and processing and handling conditions. So in this next part of the presentation, I will discuss some manufacturing considerations. There are several considerations for manufacturing drug polymer blends using melt extrusion, of which processing temperatures are perhaps foremost. Polymers process at specific temperatures which must be compatible with the active ingredient. A blend is not possible if the degradation temperature of the drug is below the processing temperature of the polymer. The screw speed will influence the highest shear rate that the formulation experiences in the process. Shear rate will influence both temperature rise and viscosity of the material in the process. It's important to control the dimensions for release rate control, since the surface area can have a strong effect on this release rate. Dimensions of the implant also influence how the implant will interact with the applicator device and affect in vivo administration, both in use and removal. Typically, multi-axis laser micrometers are used to control the dimensions of the implant. It's critical to blend sufficiently for phase, microstructural, implant-to-implant, and batch-to-batch uniformity. It's also important to melt the polymer to produce a low porosity part for drug release control, and to avoid degradation of the drug or excipients. Rheology describes how the material flows in the process. It's important to understand the rheological characteristics of the formulation and how it influences mixing, and to obtain the melt strength required to form and shape the implant as it exits the extruder. Ambient process conditions, storage, and shelf life must all be understood and controlled, since many of the materials used, particularly in the bioabsorbable polymers, are hygroscopic in nature, and it's important to control moisture content of these polymers. This graph shows the process temperatures of several different polymers, both biodegradable and bioresorbable, and then the melting point of some model drugs are overlaid on these curves. So basically this shows that how the molecules are compatible with the processing temperatures of many of these polymers. So now I'm going to talk about the implant manufacturing process. And a typical implant that requires very precise dimensional control. The material is first compounded or melt blended in a twin screw extruder and then pelletized. And then those pellets are then presented to the single screw extruder where the implant is shaped and formed. And it goes through a cooling process. You have this diameter control is that laser micrometer that I talked about, and then a polar cutter. You can produce an implant directly off of the twin screw, but it would require a melt pump. And next I'm going to describe the difference between these two processes, and that'll help explain why. So twin screw extrusion and single screw extrusion is sort of a comparison here. Twin screw extruders are star fed. The feeding is controlled using gravimetric feeding of the components. And in single screw extrusion, it's flood fed. So all the material is put in the main feed throat, and the control of the feeding is not critical. With twin screw extrusion, you have much better mixing and control of mixing because the screw design is modular and the barrels are modular. You have less mixing capability and control with a single screw extruder. You do have some mixing that occurs, but it's mostly distributive type of mixing. And the screws in the barrel are fixed, so they're not programmable or adjustable. And in twin screw extrusion, it has a very low unstable die pressure. And with single screw extrusion, it has a very high stable pressure. So the bottom line is twin screws are excellent mixers, but real low pressure, so they're not good pumps. And single screws are not great mixers, but they are excellent pumps. So you can see why both are used to create an implant. You can produce an implant directly off of a twin screw, and that's done by applying a gear pump, and I'll show how that's applied a little bit later on in the presentation. So some of the challenges in developing a process for an implant. First, the active pharmaceutical ingredients are scarce and they're expensive, particularly early on in the development phase. The APIs tend to be cohesive and have a low bulk density. Usually after they're synthesized, they go through some type of milling process, and many of them go through a jet milling process so that they can get a smaller particle size and increase the surface area and enhance the drug release. However, when you have a small particle size, the APIs tend to be more cohesive, have very low bulk density, which then leads to very poor flow properties. The implant is a highly filled material with a therapeutic duration that depends on the drug loading. So the drug loading is linked to the release rate, which cannot be too high or too low. So the drug loading, if it's too high, then it leads to processing issues and you really can't process the material. And if it's too low, then you won't achieve the desired drug release rate. So not only, it has to be balanced not only for processability in terms of whether you can actually form the implant, but even at higher drug loads, you will have more localized shear, and that's a potential for degradation. So it's something that you have to look out for when you're developing the implant. The processing parameters and scale may result in different microstructure due to particle size reduction, particle orientation, and change in part porosity. These are all things that happen in both the single and the twin screw process. As I mentioned, twin screws are excellent mixers, and they do so by imparting shear onto the formulation, and that shear can really result in a particle size reduction. So in this last part of my presentation, I will cover process options and then discuss a few case studies. So these are the process options, and you can make a pellet, you can make a monofilament, you can extrude a tube, or even some other profiled shape, and you can co-extrude shapes, you can make films. Foaming is possible if you want to control the porosity in the dosage form. And lastly, if you want to make any sort of implant that has a feature on it, then you'd have to go to injection molding. And I know we have an expert speaking today. Jim Arps from ProMed is going to be speaking about injection molding in detail. So that's beyond the scope of my talk today. So drug delivery fibers and monofilaments, you can produce sutures, precise pellets, or even coils. What's shown in the picture here is a twin screw extruder, and there's a vacuum vent, so where we're controlling the vacuum that's being applied to the process. And at the end of the machine is this melt pump. So we have an inlet and we have a melt pump or a gear pump that's increasing the pressure and stabilizing the pressure that's coming from the twin screw. And then there's a die on the other side going through a cooling device, and then the product is cooled. So I have a schematic of this process. So you have the twin screw extruder, you have a gravimetric feeder into the feed hopper, the twin screw extrusion process, and then a gear pump that's attached to the machine as shown in the previous picture. And then custom cooling devices that are used. There's air cooling, a laser micrometer for precise dimension control, and that's followed by a polar cutter. So the first case study that I'm going to review today is local drug delivery of clonidine. This study was sponsored by Medtronic, where they looked at a clonidine-loaded implant that was implanted near a single lumbar foramen. Clonidine tissue concentrations were measured at the implantation site and at incremental distances from the implant over time. And here are the results. Here you can see that the drug was detected closer to the implantation site within the first 2 weeks of implantation. And after 12 weeks, clonidine was detected as far as 4 centimeters from the implantation site. And here are the in vitro results where that shows the drug elution levels off at about 90% after 98 days. So it's a pretty long implantation period that's achievable with this implant. The next case study is about work that was conducted on a new molecule for HIV prevention, MK8591. And in case you were wondering what Dr. Anthony Fauci was doing before the COVID-19 pandemic hit, Dr. Fauci is a leading expert on AIDS, and he was quoted in this New York Times article that was published just about a year ago. MK8591 was evaluated in both biodegradable and biodurable polymer matrices. At left, in vitro results are shown, and at right, the in vivo drug release rate is shown. Consistent durable drug release was achieved in both in vitro and in vivo with all 3 polymers. For each system, the impact of drug loading on release is strong, and high loadings lead to more rapid release and drug depletion. A clinical study was conducted with PLA, PCL, and EVA. Consistent drug release in non-human primates were achieved with all 3 polymers. The figures on the bottom right shows the projected human plasma profile of the implant, shown in the green line on the graph, compared to a once-weekly oral dosing of MK8591. These images show the drug release through percolation or particle-particle contact. So you can see there's no drug release here. You can see that all of the particles in the image, and then at 18% drug release, you can see that it's releasing from the outside in towards the core, and at 100% drug release, most of the drug has been depleted. And you can really, these micrographs really show the voids that are created as the drug is released over time. So the last case I'm going to discuss today is a combination device that is made by tube extrusion. So this is a schematic of a tubing extrusion line. You have a single screw extruder here, followed by an air wipe, a support system, a laser micrometer to control the dimensions, a puller, and a conveyor. So this case is a drug-eluting ureteral stent. These stents relieve temporary obstructions in the kidney. They maintain dilation from swelling, and the result of the disease or procedure is 80% patients report stent pain. So here are the in vitro drug release rates. 3 different systems were evaluated: 7% drug, 13% drug, and 15% drug. The graph shows the drug release performance of stents containing 15% was greater than the 13% and the 7%. So the 13% drug load system was chosen for a human clinical study. Here's a picture of the stent here. It has these 2 pigtails on either end. The stents are 20 to 30 centimeters in length, depending on the patient. The control is just an EVA without any drug. And the drug polymer, again, it's EVA, ethylene vinyl acetate, with 13% ketorolac. 276 patients were evaluated, and they were looking at whether or not they used any intervention for pain. And these are some of the results from this human clinical study. These data show that patients that had the drug-loaded stent used fewer pain-relieving pills than those that had the polymer stent only. So I'm going to talk about a few other processing techniques, and then we'll wrap it up. So drug delivery via co-extrusion. These are more complex dosage forms. They're used for intravaginal rings and for other implants. The picture that's shown here is a picture of the NuvaRing, which is an intravaginal ring that's manufactured by Merck for contraceptive use. Here is a picture of a co-extrusion process. So in this case, we have a single screw that's shown on the top right, feeding into a co-extrusion head, and on the left is a twin screw. And in this case, we have a core that's the drug-loaded core with a rate controlling membrane. So here you can see this is a cross section, and we used a black colorant just to show the concentricity of the drug polymer matrix in the core, and the black membrane on the outside. So this is about 4.5 millimeters in diameter, and this skin layer is about 0.1 millimeters. Here's an overview of the co-extrusion process. So you have two extruders. You might go through a water trough or some sort of air cooling device. There's an air wipe, again, a laser micrometer, a puller, and a conveyor. Films is another dosage form, so they can be used for either transdermal or transmucosal delivery. They can also be used for implants. They can be single or multi-layers. You can use a feed block co-extrusion or lamination. Process control is critical to, again, control the thickness for the reasons I mentioned earlier. You need good dimensional controls. You have to cut these into the final dimensions using some sort of cutting device and then packaging equipment. Extruded film is collected on a 3-roll stack. So after the film exits the extrusion die, it goes through a series of rollers to control the dimensions and also cool the device. And then they're taken up on a roller further downstream. And here's a co-extrusion die. Again, you'd have, in this case, you'd have 3 layers, and this shows the flow channel, a cross section of the flow channels where the 3 different polymers would go through, and then they're formed into a co-extruded die as it exits the machine. So in summary, long-acting implants offer localized, site-specific drug delivery. They reduce the need for repeated injections and enable improved patient compliance. Development of implantable dosage forms require an understanding of materials, process, and product design. Extrusion accommodates many different material types and offer many process options to continuously produce complex dosage forms in a tightly controlled manner. I thank you for your attention, and I'll be happy to take any questions.